And thanks everybody for sticking it out. Thank you especially to the lake committees for allowing us to give this update. I have some updates on our larval fish recruitment investigations in Lake Huron. This is a CSMI project, and I'm excited to give these updates on behalf of a very large team. So I want to acknowledge just the co-authors here, and also say Ralph Tingley and John Dobeck are also PIs on this. I'll also acknowledge all the folks that helped us, jumped in the field and in the lab throughout this project for sampling, lab processing. Also like to acknowledge all the folks that helped share data with us or we got knowledge from on sampling. There's just so many people to thank. I also want to acknowledge our funding, GLRI, and the Corriganine Restoration Framework for helping out with this work. So I'll jump right into the problem, what we're charged with. This is the CSMI priority. So there's evidence for a recruitment bottleneck. That's essentially like a survival choke point for lake whitefish, larval lake whitefish, we think, in Lake Huron. So we think it's occurring in their first year of life. We also hypothesize it might be compounded by dreissenid mussel invasions, shifting climate, food web changes, especially in certain areas. And we think the impacts are different across different areas as well. So there's a lot to unpack there with this problem. But in short, we think that these fish aren't making it to a catchable size, and we think the choke point is at the larval stage. So I'll just quickly, we saw the life cycle in Andrew's talk, so he did a good job covering that. I want to quickly talk about some of the developmental stages that I'll talk about in this talk. Yolk sac larvae, we call them in this talk, sometimes called sac fry. These are larvae that are recently hatched, still have their yolk sac. I'll then be talking quite a bit about post-yolk larvae. And then I'll just go through the rest of the life cycle here and move through. But those are the main, a little bit about those post-larval age 0 juveniles as well, because those fit into some of the data that I'm going to talk about today. But essentially, the issue is we think that something is happening up here that's not leading to fish showing up over here at age 3 catchable sizes. Plenty of evidence that that's happening. So I'm going to talk about some of the sites that I'll talk about today. There's a lot of larval fish sampling that happened in 2022 around Lake Huron, but we chose 2 specific regions for some pretty in-depth sampling. Some of this will be a little bit of a recap, but I have some updates today on diet stuff as well in these regions. We chose the North Channel and Southern Lake Huron, specifically south of Saginaw Bay, down that coast, down the thumb. We chose these regions because there's a big difference in nutrient gradient there between these two. You saw on Andrew's maps, dreissenid mussels aren't very well established in the North Channel. It's also quite nutrient rich up there, more or less. And then down south here, there's a lot of dreissenid mussel establishment. The water's really clear down here. The food web seems to be a lot different. And so we think there's this extreme nutrient gradient that we could sample in just to try to help reveal some of those factors that might be responsible for that recruitment choke point. We think that they'll be more noticeable in extreme regions like this, is why we chose these. So here's some of the sites I'll talk about today. Some of you may recognize a couple of these beaches. And I'll go through, the goal and some research questions that we had when we started. So the big goal here is to partner up in true CSMI style. That's Cooperative Science and Monitoring Initiative. We partner up, we monitor as much as we can across a broad range of sites across Lake Huron to reveal these variables that might be responsible for this choke point for these fish. So we had some questions, and I'll end with these questions as well. One was, our larval lake whitefish density is going to be higher in that North Channel compared to Southern Lake Huron because of that nutrient gradient difference and less dreissenids up there. Will we see higher zooplankton densities in the north versus the south for the same reasons? Will we see better larval lake whitefish growth in the north versus south, sort of tying in there? And then higher post-larval age 0s, will we see those showing up more in the north, still in the near shore areas after the larvae grow up a little bit compared to Southern Lake Huron? And then tying those questions together, will there be less food available? Will we see that? And could that mean less food in the stomachs and then slower growth down south specifically? We did a lot of sampling. I'm going to focus on a specific one that we did in 2022, but to go through it quickly, right at ice out, we started sampling on the beach with Newston nets, try to catch those larvae right after they hatch. At the same time, at some of those sites, we took small vessels and sampled just out from the same beach at the same time at the surface, just to see if there's larval fish out there that we can catch right after hatch. This is like 5 to 15 meters total depth, still at the surface with bongo nets. We collected larval fish and zooplankton simultaneously during that time, or back to back rather. And then as the spring turned into summer and those fish grew up a little bit, we continued to collect zooplankton on the same beaches. We started bag seining to try to get those post-larval juveniles. And we also leveraged our large vessels out from some of those same beaches or nearby and across a variety of ports to bottom trawl for those age 0 whitefish as well. So we did a ton of sampling. In this talk, I have a lot of updates based just on this beach sampling. So I'm going to kind of focus in on this here. We can chat about the other stuff later though. So focusing in on beach sampling, we drug the Newston net, we collected larval fish, and we drug a zoop net. We collected zooplankton to see what was out there to eat. We identified and counted everything. For the fish, we did genetics to confirm species. Did some morphometrics on them, kind of poked at them quite a bit actually. Did some aging and diets on these fish. I'll quickly cover some trends with just densities here so we can get into the growth stuff and diet stuff. But one big thing, at least at our sites, from north to south, the beaches I mentioned, we saw an order of magnitude more larval whitefish right on that beach in wadable water compared to just off the same beach or along the same time frame. So that was, I like to say that life's a beach for these small fish, and it seems to be true in some sites. They really need that beach. Same trend with zooplankton. We saw an order of magnitude more right on the beach in that early springtime compared to just off the same beach at the surface anyway. Order of magnitude more zoops detected in the North Channel compared to those southern Lake Huron sites, which is something that kind of fit with one of our questions there. Larval whitefish densities, however, were kind of all over the place. They busted a couple of our questions. I'll get into that. They're all over the place. And we saw that high beach larval lake whitefish densities didn't always mean high zooplankton densities at that same site. Those were kind of mismatched a lot too. I'll have a graph here that touches on that. So here's the overall mean densities for those sites I mentioned in the north versus the south. You can see they're kind of all over the place. And Harbor Beach down south definitely surprised us there. They had the highest, that beach had the highest density for larval lake whitefish across the sampling season compared to all the rest. And if I take that fish density graph and just shrink it and put it on top of the same zooplankton density graph, this is just mean zooplankton, the number per liter of water stacked below the fish densities. You'll see right away that it didn't always match. So high zooplankton density at certain sites didn't necessarily mean we saw high whitefish densities, and vice versa. You see Harbor Beach highlighted had very low zooplankton densities there in general, but very high fish densities. It's a little bit of a red flag. Looking closer at these sites, I'm going to talk specifically about two right now, Fourth Sand Beach in the north and Harbor Beach down south. The reason why I want to focus on these two is because the densities across time for larval fish, larval lake whitefish, were pretty similar. The fish sort of started to peak up on the beaches and then peaked at about 1 fish per cubic meter and then tapered off as they got bigger, probably avoiding the Newston net at that point. Both sites are pretty similar. Harbor Beach was a little higher at their peak, but similar density patterns. Yet when we looked at the length of those fish each week, you'll see that over the same time frame, the North Channel fish tended to get bigger until they disappeared from what we saw, and the Harbor Beach fish never really did get much bigger until they disappeared. And that could be explained. There's a lot of ways to explain that. The Harbor Beach fish might be moving out more and new populations are moving into that beach. We don't know exactly why, but it's definitely grounds to investigate these two sites further, and especially in terms of growth. So now I'll show a bunch of these growth plots. I broke these out, so I'll just explain these for a second. I broke them out to yolk sac fish only first, and then I'll show post-yolk fish, and then I'll just combine those two developmental stages into one plot. The blue rings are the North Channel site. The orange, a little bit smaller rings are Harbor Beach sites. And this is just the total length of the fish versus our estimated age in days by using their otoliths. So you'll see, I mean, not too surprising on this one. This is a really narrow snapshot in time. It's like the first week of their life, more or less, a week or 2 weeks. And you'll see that they kind of overlap. There's no big, they're kind of messy. Generally, as the fish are getting bigger, we're estimating they're getting older in days there. But as you look at the post-yolk larvae only, you can see that Harbor Beach is starting to fall below North Channel fish at this point. So they're still getting bigger with their estimated age in days, but they're not getting, at the same age, they're just not getting as big as those North Channel larvae were. So when you combine everything, you can see it's still pretty messy. Again, pretty narrow snapshot in time for these estimated age plots, but you can see that Harbor Beach starts to fall behind. Another thing we looked at, instead of counting the increments on the otolith, we just looked at max diameter of this otolith, of the fish. It's a good, quick way. We thought maybe be a nice proxy to compare, to look at growth. And so we took a quick measurement of the otolith, and even here in yolk sac larvae, you can see by these data that the Harbor Beach fish seem to already be falling behind. Even as yolk sac fish, their otolith is getting bigger, but their body's not getting as long as those North Channel fish. So it raises a lot of questions. And of course, post-yolk larvae look similar, falling behind down south. And as you combine those plots, they look similar to the length at age plots. Here's an otolith from a larvae. I want to quickly show this. I'm not going to talk too much about aging, but we essentially, we count these increments and consider them roughly days. But one thing it's broken too, I want to point that out, but it's still beautiful, I think. Beautiful but broken. And you can see that there's some really good counting windows here. But one thing I want to just quick point out is that you see some thicker increments. In the literature, these are usually described as check marks. We need these to know where to start counting. They're really important, but according to the literature, they can happen through several different types of stressful events, probably more than we know. They happen when a fish hatches from an egg. When a fish absorbs its yolk sac, you usually see a dusky double band. You'll see some other ones. If a fish goes through a cold shock or starves, you'll see these. It's a slowdown of a day or two of growth, creates a thicker band. So we count those in all our fish. So if you look at check marks per otolith overall, from the North Channel site to the southern site, you'll see that it's way more variable down south. They tend to have their check marks, even as a yolk sac fish, we'll start there. They tend to have, their check marks are just more variable. You should see about 2 or maybe 3, but we see more or less. It's just all over the place. Post-yolk fish, they tend to catch up with each other. They have about the same amount of check marks. And then you'll see when you combine all larvae, it's really driven by those yolk sac fish going through something in those first couple weeks of life, possibly. Get into diets. This is just the number of things we found in their stomachs. And even yolk sac fish will eat. So I think that's kind of interesting. I'll show the North Channel fish in blue again and Harbor Beach in orange. And the axes are kind of wonky, but that's just to compare to the post-yolk fish eventually here. But you'll see that most of the time their bellies are empty. It is interesting that North Channel fish, as they start to get a little bigger, they start to, even as a yolk sac fish, they'll start to have more things in their belly. They start to eat a little bit more so than Harbor Beach. Post-yolk fish, the North Channel fish really start to take off with items in their belly compared to southern Lake Huron fish. I think even, as a post-yolk fish, we still found some empty bellies down south, which is a little bit of a red flag there. When you look at what they're eating, and look at the means of those counts for specimens in their gut. It's mainly, as yolk sac fish, there's not much here, reasonably. They have a yolk sac still, but they do eat. North Channel fish eat the cyclopoids. They tend to eat Holopedium, which I thought was really interesting. Little gooey zooplankton in a gel packet. I don't know how I don't know what they do with those, but that's very interesting. Bosminids, sometimes you see some calanoids. Southern sites, it remained pretty low. You don't see that cyclopoid, they don't see those yolk sac fish targeting cyclopoids as much. When you look at post-yolk larvae, this is when they really should be eating. You see that right away, the North Channel fish, they just have a lot more of these things in their stomachs compared to down south. Still, the cyclopoids, bosminids, and to some extent calanoids tend to be a pretty good item that they tend to seek out and eat for zooplankton, general groups anyway. If you look at the length of those zoops that I just mentioned, the top three I'll talk about that they tend to target, we measure those zooplankton inside their, once we dissect them out of their stomachs. And you have to kind of squint at this one a little bit, but you can see that Southern Lake Huron, those three taxonomic groups tend to be a little bit bigger. It's noisy, but they're a little bit bigger in the stomachs, which is a red flag to us. We saw that and thought, well, maybe they're forced to eat. Maybe there's a mismatch there, and the zoops are a little bigger down south, the ones that are down there. So they're forced to try to eat these bigger ones. So if you look at the mean length of those zooplankton in the wild, again, those same three, first thing you'll notice is we have some missing data here probably, and that data is, it's counted, they're measured, it's just not in the database yet. This is where it really gets fresh off the lab bench here. But what you'll see next probably is that the zoops are really big right out of the gate down south. Compared to up north, these are a lot bigger. I don't have the error bars on here yet, but they're just a lot bigger in that critical moment right at ice out when you probably have larval fish needing to eat. We also take the gape on all these larval fish that I've talked about, and that's just the amount, the width that, or sorry, the maximum measurement for their mouth to be able to open. It's still in millimeters, and it's still across the same time scale, so I can kind of overlay that data right onto the zoop size data, just to look at what kind of zone they're in here for trying to be able to eat these things. It fits right on the same scales. And so the gape for North Channel fish tends to fall right within or a little bit above the size ranges of their favorite food items there. If you put that same gape measurement for the Southern Lake Huron fish, they're down within the sizes now. I mean, this must be a little bit more difficult for them to eat these zoops, is what we're guessing. So it's another potential line of evidence for a mismatch, and it's size related there. One thing I want to quickly point out too is the amount of plastic we find in these fish. It's really alarming. We weren't targeting a plastic study here, but we did, for each fish, we didn't quantify it, but we did say yes or no, there's plastic in it. And you'll see right away the Fourth Sand Beach, the North Channel fish, about 90% of those post-yolk larvae that are eating had plastic in their bellies. So just really interesting there, kind of scary. Something to explore further. I'll quickly point out the next phase of that field season was to start trying to catch these post-larval fish that usually, in some cases, still hang out on that same beach and utilize that beach nursery habitat. So we did a lot of bag seining. This is where our multi-agency map shows up, and this is really useful. I'll just quickly say the densities are all over the place for these as well. But one thing that's notable, these X's are kind of small, but we caught none in our Southern Lake Huron sites. We were able to catch these up north, around Lake Huron and various ports and beaches, but we didn't get a single one at any of our sites after getting all of those larvae, especially at Harbor Beach. And we don't know where they go or what happened to them. They could be moving out in this case, and they just don't stay on the beach. Could be moving through the system or out to Saginaw Bay. We just don't know. So I'll just kind of recap some of these research questions. The higher density in the north versus the south, that one we got busted on, not necessarily. Harbor Beach showed a really high whitefish larvae density. Higher zooplankton densities, north versus south, yep. Better larval growth, we are seeing that at least in the two sites I've shown you. So it's a small start, but we are seeing that at certain sites. Higher post-larval age 0 densities in the north versus the south. That wasn't necessarily true because of the multi-agency effort. We saw that there's really high densities of these post-larval juveniles or age 0s out from Alpena there. But we didn't catch any down south. So again, that was just kind of alarming there. Less food means less food available, means less in the stomach and slower growth. That evidence is certainly stacking up for a yes down south, at least at one of these sites. It just seems like there's a mismatch there. So I think I'm getting pretty short on time. I'll just leave it there and ask if there's any questions. Thank you.